Good afternoon, ladies and gentlemen. My name is Petter Hartman and I'm the CEO of Medicon Valley Alliance, and it's a pleasure for me to welcome you all to today's MVA Oncology Network meeting. This meeting is organized in collaboration between Medicon Valley Alliance, of course, and our friends at Lund University, Copenhagen University, and our sponsor, Höjberg. Before we start, I would like to take you through some practical things. You're automatically muted while joining this meeting. If you have any technical questions, please use the chat. But you can also ask questions to the panelists during today's program, and we will summarize them at the end of the meeting and bring them up. Please, if that's the case, use the Q&A function for questions. And then please also note that this meeting will be recorded. For those of you who have signed up for this event, we will send out an email tomorrow with the presentations and also a link to a survey. And we would really appreciate if you could take the time to answer the questions in the survey to help us to improve, which would be very much appreciated. The Oncology Network is one out of, in total, 5 networks that we at Medicon Valley Alliance organizes. And as a member of Medicon Valley Alliance, you can attend all meetings free of charge. Besides the Oncology Network, we also have a MedTech network, an Executive Club network, a Microbiome network, and also a R&D network, which could also be of great interest for several of you today, I guess. If you have any questions about the membership and how it works and how you can attend or even host a meeting, then you should, of course, be most welcome to contact me or one of my colleagues afterwards to get this information. So let's switch focus to today's program. We have a host of today, Lund University, and we have invited Johan Malm, professor at Lund University, to help us guide us through today's program. Johan Malm will also be introducing the topic today. And Johan, if you light your camera and unmute, and then I would like to leave the screen for you, Johan, and you can continue.
So, good morning, everybody. I hope you can see my screen now.
Yes, but not in presentation mode, Johan. So please.
Presentation mode. No, it's.
There we go.
I hope. Hi, everybody, and you're very welcome to today's meeting. I'm glad that you've come. And before we start with the presentations, I would like to say a few words about cancer statistics and the cost of cancer care and precision medicine. According to the WHO, it's expected that about 1 in 6 deaths that occur will be due to cancer. So this makes cancer second to only to cardiovascular diseases. Now, there are many risk factors that contribute to this, lifestyle and environmental factors, and also age. And here you can see a graph that is very clearly illustrates the fact that as you get older, the risk of being diagnosed with cancer increases. Now the life expectancy has increased in many countries, and the demography changes. More and more people belong to the older age groups. And in Europe, this is what is expected to happen within the next 80 years. As you can see, the two age groups in pink and orange representing the population over 65 years of age is expected to increase by more than 50%, and this will result in a larger number of older people who will develop cancer, and this will of course be very, very expensive. As illustrated here, you can see the total cost of cancer per capita in different European countries. And the total cost 2 years ago was approximately 200 billion euros in Europe. And this is not just treatment, but also lost years of working and sick leave and other costs that contribute. So cancer caused nearly equally large costs within and outside the healthcare system. And so health spending on cancer has been increasing much faster than cancer incidence. Now, not only the cost, but also the death rates differ among the different European countries. As you can see here, with a standardized rates of death in different European countries. So with this background, it's obvious that the pressure on our healthcare system can be expected to increase also when the present pandemic is over. And since the cost of cancer care will increase, it's obvious that we need improved diagnostic methods and improved treatment. Now, this takes us to precision medicine, also sometimes called personalized medicine. Now, there is no clear definition of neither of these two terms, but they are often used interchangeably. Personalized medicine is the older term, and there was concern that the word personalized could be misinterpreted to imply that treatments and preventions are being developed uniquely for each individual. In precision medicine, the focus is more on identifying which approaches will be effective for which patients based on genetic and environmental and lifestyle factors. Now, the first example of precision medicine is more than 100 years old, and this happened in Vienna in 1901 when the ABO blood group system was invented or developed. And they realized that each patient's biology actually impacts the result of the blood transfusion. Some of them were successful and some were deadly. Now, for many years, not much happened in the field of precision medicine. Cancer has been treated with surgery and radiation and chemotherapy, more and more advanced and in different combinations, and with obviously much improved results, but in still limited success or even failure in many cases. Now, the first targeted cancer therapy was approved in the 70s, so that's almost 50 years ago. That was tamoxifen, which is an estrogen receptor blocker. So you could inhibit estrogen-dependent cancer cells from growing. And then with the discovery of oncogenes and tumor suppressor genes and the sequencing of the human genome almost 20 years ago, we now have a basis for understanding molecular mechanisms leading to cancer. And we also have made possible the development of a large number of the new targeted therapies. So we have the possibility to move from one size fits all approach to a more an approach of personalized medicine, being proactive instead of reactive. It's illustrated here in an article in Nature, which emphasizes the fact that it's not just diagnosis, but also and treatment, but also prevention. So with the use of artificial intelligence, different types of omics information and phenotype information will ultimately benefit patients and also society. And with this short introduction, I would like to ask the first presenter to begin. And this presentation will be given by Jens Victor Nørgaard, who is a European patent attorney at Højberg, which is a law firm that provides services within the fields of intellectual property rights. And Jens Victor will talk about the value of IP in personalized medicine. Please, Jens Victor.
Thank you very much. I hope you're seeing my screen now. Can you confirm that? And the presentation?
Yes.
Great. So thank you very much for the introduction, Peter and Johan. I will just be talking briefly, sponsors of the event, about what you can do with IP in this field. This is a very important field where treatments are more personalized or more precise than when you just treat everybody with the same kind of treatment. This is just a slide with my CV. So Højberg is a patent law firm. As Johan said, we prepare patent applications and patent strategies for companies and researchers. We have a very large IP, life science IP group with 25 life science attorneys located across Scandinavia, Stockholm, Lund, Copenhagen, Malmö, and Aarhus. So looking at the typical chart of what can be patented, we have our products, we have formulations like compositions, you have downstream processes, you have upstream processes for making these products, and you have downstream processes for use of the products. And this is not where the place that precision medicine comes in will be here on the downstream uses very often, because we're not very often not talking about new products. It will be more optimal uses of known products. And in many cases, we will be in a situation where there's maybe not any exclusivity left on the market. This is a typical slide of how you can look at the exclusivity of a drug. You have a first patent application covering the product. You maybe have a second patent application covering some medical uses. You have a market entry at some point in time. And after that, you have data protection in US, data exclusivity in Europe, excuse me, EU up here and the US down here, and maybe orphan drug designation, which you also get for some cancer types. And very often precision medicine comes in at a later stage in this life cycle of a product that's on the market. So very often you'll be close to a loss of exclusivity where you face generic competition. And this is where precision medicine comes in handy to permit value extraction by codifying what you've found, filing a patent application on it. It enables raising funds for clinical development if you form a company, but you can also, it also permits value extraction through license agreements, and you can delay the loss of exclusivity for drugs, if it's a drug-related invention, of course, because as Johan said, precision medicine can be used both in prevention, in diagnosis, and in treatment of cancer. And what can you then patent? You could patent biomarkers before or during treatment, following the efficacy of your treatment, or selecting particular subgroups of patients for treatment. Personalized dosage regimen, often in response to anything you measure in the body. Ex vivo stimulation of cells could also be personalized. Drug combinations can also be personalized medicine, and different ways of overcoming drug resistance can also be regarded as precision or personalized medicine. So in order to get a patent, what is required is that there is some kind of a technical effect that will often be in the form of a patient benefit. It should always be to the benefit of the patient, otherwise it doesn't really make sense. That could be in the form of fewer side effects, higher efficacy, identification of responders in a subpopulation, improved efficacy, and whatnot. So this is just a very brief introduction on the possibilities for protecting new findings in precision medicine with patents. Thank you for your attention.
Thank you very much, Jens Victor. This was interesting, and IP and legislation is obviously very important for all of us when we get new, interesting, exciting results that can be turned into products. Now I turn to the next speaker, who is Yuri Marco Varga, who is Professor of Clinical Protein Science and Imaging at Lund University, and Yuri will present the European Cancer Moonshot Center in Lund.
Thank you, Johan. So, Peter and MVA, thank you for inviting me for this afternoon Cancer Network interaction. And funny enough, you know, I'm right now at the affiliations you see in the first slide here.
Georgi, sorry to interrupt. Can you take over the screen and share your screen with us? Or maybe we should ask Jens Victor to stop share, and then you can start sharing, Georgi. So if you start sharing your screen now, Georgi, it should work.
Okay.
And in presentation mode, please.
Can you see it now?
It's not in now it's in presentation mode. Thank you.
Okay.
Go ahead, Georgi.
Okay, so I was just saying that you know, I spent almost 20 years in AstraZeneca, so I'm now in the academic and clinical area right now. But with the background, I will introduce you to a very exciting area that is currently ongoing. And it started, so the whole cancer moonshot activity started by a thing that happened to the former Vice President Joe Biden, his son Beau Biden. And from there, it actually took off, and it became then the Cancer Moonshot program. And the whole program is really about accelerating, being very close to patient treatment. There are something like 45 different clinical centers in the US where screening and prevention is the focus of activity. So it also enforces a more cooperative and collaborative effort. And here they actually have been able to include also the pharma industry, biotech, governmental institution, and academia. And one of the big things about this program is really to try to push in order to share data globally. So this is a view from the first sort of globalization and meeting that took place with Australia. That was the first country to join in outside the US. So it's a 7-year program. Still, it's past halfway through, and it has a quite significant budget. And what has happened here is that although now there has been a change from the Obama administration to the new administration, there hasn't been any sort of deviation from the plan that was originally set. And Joe Biden is still the head of this program. Now, there is one part that relates to the molecular sort of phenotyping of cancer diseases, and it's shown on this slide. So, and we are partners here, and you actually have then 13 countries, and it also captures an N number of 75,000 patients being screened through in 15 different cancer types. And there are now 33 different type of institutions in these 13 countries. It's a very active and interactive consortia work where genomics, proteomics, transcriptomics, metabolomics are now being profiled in various cancer diseases. Now, in Lund, we established the Cancer Moonshot, European Cancer Moonshot Center in Sweden, here in Lund. And you can follow us on social media. We are pretty active and a lot of things are happening. We also had last year a link to Brazil, where we were able to establish a partnership with the National Genomic Center in Brazil, in Rio de Janeiro. So we have also resources with partnership with Brazil. So we're going a little bit outside the European remit in terms of geography. So this works extremely well. We are very excited, and we just recently met up with Joe Biden at a Cancer Moonshot meeting in Dublin. And here you can see you and myself and Roger being in interaction at this meeting summit. So what the way it's working is that there is a strong interaction on a national level, and it's actually headed by the NIH. So NIH is the coordinating organization unit, and NIH is then linked to the leadership of Joe Biden and his team to lead the whole program forward. In Lund, we have set up a pretty impressive proteogenomic center. You are very welcome to visit if you're passing by. And we have top-notch, state-of-the-art, high-capacity screening platforms. So today you can actually do a lot of things that you were not able to do just a few years ago. And you see the numbers being put up there. Another important part is that we also have been able to get the national infrastructure of medical protein science included here. And we can then interact on a local level together and bring forward developments. So the whole sequencing by mass spectrometry, where we are kind of moving in on the clinical protein area, is something that it's not new. I mean, there has been a number of Nobel Prizes in the past, over more than 100 years. And the principal basis of the sequencing capacity is something that is still very much in fast progress. So in the protein area, to be able to analyze, sequence, and determine modifications, you actually do not have an opportunity like on the genomics area to multiply, to use a PCR technique. So there are no PCR techniques for proteins, and that somehow gives it a certain position where the experience and the know-how and really cutting edge is key in order to be able to provide high-end data. So here I put in a few slides from an open house event we had last year, where you see visitors coming. So we had more than 400 people visiting over half a day. There were like from high schools, there were some sports clubs that came visiting, and also just general people around in society and in the community. Now, you can actually do sequencing and protein analysis in many different ways. You can today multiplex bigger numbers of patients, so you can start to go on larger sets as well. So that means in the hundreds and now pushing up towards thousands of patients or analyzing proteins. Now, a very important thing happened just a few weeks ago, and that was actually after the human genome. That was the mapping of the human proteome blueprint. So that means that all of the proteins coded by the human genome have now been established, and now that is available for everybody to use. And so here you can go in, and just as with the genome, you can actually assess, you can look for different protein, and the variety here is really the functional aspect or functional genomics, if you want. And many of these different principles, platforms, methodologies, workflows we use at the European Cancer Moonshot Center and can be set up in many of these different activities where we are involved on a global level. Now, one aspect that is very important is, of course, the treatment, as Johan alluded to in the beginning, and that is the treatment of patients and how you use all of these data. And it relates, of course, back to let's say, classical pharmacology, but it also has to do with the tumor heterogeneity that you need really to fight against. And here we have done a lot of pathological work together with pathology in Korea, in Hungary, and all over Germany as well, the Netherlands, in order to try to understand more of the details of what's happening within the tumor environment and also within compartments. So you have to understand it's not one type of cancer cell, but they are many different types of clones, and all of them, they have different roles. Now, when you start to administer drug to patients on an individual basis, these different clones will react differently, and you will have then challenges that relate to the patient, where you will see resistance built up, where you have metastasis being developed, and sort of a regeneration of the tumor as such. So we have been leading in Lund in the area of being able to map the localization of drugs. And here I just show a few images that were published some years ago. And we are currently also collaborating with drug pharma industry in order to characterize different type of drugs. So with that, I would like to just acknowledge many of the providers of financial support and also other types of support. And here especially I would like to mention not only the NIH, but also IKEA and the Kampat Foundation, who has been very, very instrumental in order for us to be able to set it up and build it. So with that, I would like to thank you for your attention.
Thank you very much, Yuri. Interesting and exciting. And I think we go on with the next presentation, who will be given by Eva Hoffman, who is professor of molecular genetics at the University of Copenhagen. And Eva will present whole genome sequencing, mutational signatures, and interventions in cancer therapy. Please, Eva.
Okay, thank you very, very much for the invitation here today as well. In the lab, we're interested in genome evolution, and we work in spaces that are typically to do with pediatric, but also health in embryos and also in our germ cells. And increasingly as well, we've also now started with the advent of data sets coming out with whole genome sequencing to go in and start looking at mutational signatures and also what this might mean for interventions in cancer therapies. So I'm just gonna show the, okay, my thing is stuck. Here we go. That cancer genomes are complex, they evolve. This is a typical feature of evolution. And currently we're seeing an explosion in the whole genome sequences that are becoming available, in particular with the Pan-Cancer Analysis of Whole Genomes Consortium, the PWAC, that this year alone have published over 15 different papers on thousands of different cancers. There are different types of, they're well stratified now clinically. They're coming from all over the world as well. And they're really allowing us to start looking at mutational signatures, but also, of course, mutations that are occurring in tumors over time, but also potentially in germlines and also somatic driver mutations. One of the advantages of getting a whole genome view is that it doesn't just focus on trying to find drivers where an exome mutation might have taken place, but it also allows us actually to go in and look at the type of different genomic changes that are taking place in time as well, and particularly, of course, in response to treatment. So mutational signatures first really hit the limelight in a very large scale fashion, at least. Originally after the APOBEC signature was discovered, but also much broader here in 2013 from Peter Campbell and Mike Stratton at the Sanger Institute. And what they report here, for example, are the different somatic mutations in different cancer types shown up here. And what they managed to do as well was also start looking at, of course, what type of mutations were accumulating in the cancers. And what they came up with was using some very, very elaborate algorithms were mutational signatures that were classified in this case here. And we now have another 20 at the very least that have been elucidated as well. And there are some signatures that we kind of expected would come. If we look at, for example, the signature 6 here for DNA deficiency in mismatch repair proteins, this is very well known that if you can't spell check your DNA that's been replicated, particularly mutations in microsatellites, they accumulate in the genome. And these were found not just in the colorectal cancers that were looked at, but also somatic mutations, say in melanoma and also in head and neck cancers as well. So quite unexpected. Other mutational signatures that came were the BRCA1 and BRCA2 mutations that we know from hereditary breast and ovarian cancer predisposition, the APOPEC signature as well, and then also some age-related signatures, some that were also seen in smoking, started coming as well. One of the things that I think this illustrates is that we can start doing new discovery with regards to the mutational signatures, both as a validation for somatic or germline mutations that we know of, or perhaps also complete new exploration. And this is an example from Lynch syndrome, which is Lynch 1 and Lynch 2, which basically are due to mutations in mismatch repair. Classically, they are germline inherited in an autosomal dominant way, basically. So a single allele from one of the parents is deficient, and it's high penetrance, meaning 80% or more, certainly for colorectal cancer, before the onset of 40 years. One of the things that characterizes certainly the colorectal type is a high degree of microsatellite instability, or MSI, and this is generally speaking measured across a panel of different markers in the genome. The minimal one is 5, but there's also expanded panels now. However, If we start looking across the different cancers that we see in the different families that are predisposed to Lynch syndrome, particularly Lynch 2 syndrome, which includes cancer outside of the colon, what we see is that the association of MMR and activation, though we don't see it anymore in immunohistochemistry, we start seeing different degrees of microsatellite instability that show high instability, for example. And for example, in breast cancer that are associated with Lynch syndrome, you can see here that the microsatellite instability here is actually quite poor in these cancers. So why does this matter? Well, we think that perhaps the disease itself and how it evolves might not be the same, depending on which cell type we're in and which cell type one loses mismatch repair activity. But another possibility is also potentially that there are mutational hotspots in different satellites. And if we could probe them genome-wide, we might actually see that perhaps different cancers have different MSIs associated with them across the genome. And this would allow us to have improved diagnosis. So for example, we wouldn't use temozolomide or platinum compounds for treatment because MMR mutants or cells that are mutated are resistant to these compounds. We would also be able to start identifying driver mutations that might be important both in tumorigenesis and also in drug resistance responses. So for example, 30% of colorectal cancers, they have mutations in PTEN, which is thought to be one of the drivers in the colorectal type cancers within the MMR family. And then one of the, I think, very exciting things is that we know that MSI high, so a high degree of mutations in these microsatellites, they correlate with good outcomes for PD-1 treatment, possibly due to a high level of frame-shift mutations that generate peptides that are not normally produced and then presented to the immune system. And I think it would be very, very interesting to see whether we could use genome-wide MSI rates or mutational signature to try and see if we could find cancers that might be particularly vulnerable to PD-1 treatment. One of the things that is also coming across in the various different cancer studies and the mutational signature studies is, of course, that different cancers have different mutational signatures. And I think that's not surprising from what we know about certainly the germline predispositions to these types of cancers. But it's very good because it means that we can use mutational signatures as a validation for specific germline mutations that we see, but also, of course, somatic mutations that we have a poorer understanding of. One of the things that this hopefully will allow us to do is to find mutational drivers and also, of course, understand tumor evolution that would allow us to really benefit and really benefit from our knowledge of synthetic thickness and lethality in treatments. So basically exploring the therapeutic opportunities within, for example, in this case, a DNA damage response. There are more than 750 genes in here. And actually trying to figure out from just expression studies what's active and not is very difficult. And having a mutational signature would help us, I think, guide a better precision treatment. So the way that synthetic lethalities work is basically that one has a hit, for example, in topoisomerase 1, and that means that a second gene, in this case here, PBMR1, becomes very, very important for life, a cellular life. And it means if we can identify cancer stem cells, for example, with mutations in TOP1, we could then, for example, if we have the right drugs, we could start targeting these synthetic interactions, so that the two hits, both of them, would be vulnerable, obviously, to curing cancers or improving outcomes in response to treatment. And these are the kind of synthetic lethalities that we know very well from a range of different screens. There are over 50,000 papers out in the literature about exploiting synthetic lethalities in the DNA damage response. And I think the rate limiting steps really here has been to understand which genes are actually active in the various cancers, and using mutational signatures hopefully will help validate expression studies, for example. So I just want to thank the people in the lab, and particularly Simon and Jakob, with whom we work to try and exploit and understand genome evolution in cancers. And thank you very much for your time.
Thank you very much, Eva. Very impressive presentation. Martin Johansson is the next presenter. He's a professor of clinical pathology at Gothenburg University and also affiliated with Lund University, and Martin will talk about challenges in cancer pathology. Please, Martin.
Let's see, Martin, can you unmute, please, and start your video as well?
Or, sorry about that. Now I hope you hear me. Yeah.
Okay.
Okay, thank you very much for having me. And talking of challenges in cancer pathology in just 10 minutes is quite a challenge in itself. So I have to resort to a couple of examples from the current situation. So clinical pathology forms still the basis for diagnosis of all diseases where some kind of changes, disruption in tissue architecture is seen. And the bulk of diagnostics is carried out with traditional formalin fixation followed by paraffin embedding and microscopic analysis still. Molecular pathology is on the rise, and today tissue is analyzed with gene panels, with sequencing, also with PCR. Whole genome sequencing is a coming thing, which will be performed more and more as prices drop, with added steps of in silico analysis, mainly of genes of importance for disease management. And this has proven to be of great importance for genetic disease or for treatment decisions based upon genomic changes, for instance, like in melanoma, BRAF mutations, activating mutations, and EGFR activating mutations. So this is becoming more and more important. The current surge, however, of targeted therapies increase the demand for development of predictive markers to identify patients that would benefit from a certain form of treatment, like checkpoint inhibition, for instance, or anti-angiogenesis regimens, RTK inhibition treatments. More and more targeted therapies increase the demand for tailoring. And this is encompassed by the buzzwords personalized medicine in a way, or precision medicine for that matter. So novel targeted therapies are not only expensive, but also of potential harm for the patient. And many of the current studies going on in that respect does not always start with genomic alterations, but analyze instead RNA sequencing material from histologically derived mRNA. But one thing there is that proteins are the units of biological function, whereas RNA is a unit of potential function, and that is at odds with sequencing of genes, for instance. So it's not always that the RNA expression is translated, of course, into proteins with actual properties in the tumor. And I think the major challenge today is to integrate findings from sequencing of RNA into a histological context. And here pathology must serve an active function. So I've chosen one example here from ESMO this year, just to illustrate this. This is a very, very ambitious study, studying kidney renal cell carcinoma, where they look for a predictive value for response to the checkpoint inhibitor nivolumab. So it's a great big consortium, and what they do is they have the NivoRen. It's a classical trial of nivolumab, and they have blood immunophenotyping. They have also derived gene expression signatures from formalin fixed and paraffin embedded material, classical histological material. On these transcriptomics, they apply two signatures. They have a signature called T effector and angiogenesis signature, and a kidney clear cell renal cell carcinoma immune classification that they developed themselves. This first one here, the T effector and angiogenesis signature, is picked from a previous paper by McDermott that used it in a similar setting. And the McDermott paper looks like this. It concerned also a treatment where they tried to stratify the patients and gain signatures from, again, RNA. What they do, and which is a classic today, is that they interpret bioinformatically the RNA signatures, and they derive a signature for angiogenesis, for immune cell, and for myeloid inflammation based upon transcriptional levels of various RNAs. This study from ESMO also incorporated a study from Becht and his team who used a similar approach again, but this time they looked for transcripts specific for 10 types of immune cells. And immune cells are, of course, extremely important for the targeted therapy of checkpoint inhibition, for instance. There they picked up a message from these cells, specific messages according to each and all of these inflammatory cells. Based upon these results, they get signatures like this one, angio low and T effector cell high, that respond well to the therapy, the nivolumab therapy. But the angio high, the T effector high, respond less well. And you have other groups here as well. So that means that T effector high, that's the same. Angio high there has a lot less of response than the angio low. One could also see here that it distributes according to these signatures. It's a conundrum on how this translates into the tissue of the patient. And they also make a signature of their own called KICK, where they identify an immune high stromal low signature in two phases here. Also, again, according to the expression of various RNA signatures. Not surprisingly, perhaps, these two, the McDermott paper and the current ESMO presentation, they to some degree overlap, but not to a complete degree. One addresses a stromal low feature, whereas the other has an angio low feature. So why don't these signatures overlap, and why could it be that some tissue interpretation might be adequate in to bridge these gaps? Well, I'm showing you here 6 cases of clear cell renal cell carcinoma. It's double course. And as you can see from this low magnification, there is a tremendous difference from, for instance, here is one case of renal cell carcinoma, here's another case. And you can see also without glasses that there are major differences here. This is a classical renal cell carcinoma with very little stroma, a lot of clear cells from the cancer itself. Here you have another type of renal cell carcinoma. Still, it's a clear cell renal cell carcinoma, but it has more of a stromal component. This is another type, a lot of capillaries, a lot of angiogenesis going on here. And here you have yet another version of clear cell renal cell carcinoma, the eosinophilic variant. This is a biphasic renal cell carcinoma, and that shows an area here that is of eosinophilic type, and you have another area over here, which is of a more classical clear cell renal cell carcinoma type. You have vessels here. Are these vessels equal to a stromal compartment? Perhaps. You have also a vessel over here that can be, and all these vessels can be stained for CD33, which is a renowned vessel marker. And you can see that the vasculature is very densely knit in renal cell carcinoma. But you can also stain for macrophages, for instance. By staining for CD68, you can see here scattered macrophages. So these are from area B of this biphasic renal cell carcinoma. You have scattered macrophages here and there, diffusely scattered through the tumor component. But in area A, it looks like this. You can identify macrophages here and there, but you also appreciate that the cancer cells themselves seem to express by immunohistochemistry protein for CD68. If I increase magnification, it looks like this. You can see here that the cancer cells are quite high in CD68. If you were to look for the transcriptomical levels for CD68 as a readout for myeloid cells, the question is, where does that signal come from? Is it an innate property of this type of cancer that we see in area A that might load with mRNA 468? CD3, this is of importance for immuno-oncology. You can see that in area A, the CD3 positive T cells are outside of the cancer compartment. In the stromal compartments, they are standing there, excluded from the cancer, whereas in area B, they are diffusely spread across the cancer tissue. If one were to look for RNA in these cancers, I would assume that the message levels would be approximately the same, but the distribution, the difference in distribution must mean something in this context. So, summing up, I think that it's safe to say that pathology will, for a long time to come, be the golden standard for diagnosis and grading of malignancies. Molecular pathology is a coming thing, offering important services in the diagnosis of mutations. But much of the research into current treatment prediction addresses mRNA levels in tumor tissue. And here, I would believe that histopathological correlation is an important challenge. And the histological site for actual protein expression in tumor tissue is probably of highest importance for the correct interpretation and of high impact for development of predictive measures. So having said that, I have reached my time, and thank you a lot for this.
Thank you very much, Martin. I'm glad that we have clinical pathologists, because these microscopical images are really hard to interpret. The next presentation will be given by Krzysztof Krawczyk, who has a PhD in clinical pathology, and Krzysztof is the project manager at the Lumito, which is a company with a patented technology for improving the diagnostics for tissue samples. And Krzysztof will talk about tissue diagnostics of the future. Please, Krzysztof.
Yes, thank you. Could you also please confirm that you can hear me and see the slide?
Yes, it's fine.
Thank you. So thank you for inviting me for this very nice meeting. And as you mentioned, you can see two titles on this slide. You can probably guess which one was prepared by our PR department. I prefer to go with the upconversion nanoparticles as a tool for tissue evaluation, because this is what I would like to tell you about. Company Lumito, where I work, is a spin-off from a research group that was led by Lund University professor Stefan Andersson Engels at Atomic Physics. We adopted our current business model in 2017 when we decided to develop our own products instead of licensing the idea. And we are working on a novel imaging technology that is designed for digital pathology, and I'll tell you about it in a moment. And we are currently moving from more of a research phase into commercial phase. The reason why the company was founded is more and more need for tissue diagnostics, and there is an estimated 10% increase, annual increase in the need for a number of tests that need to be run. Now I think newer estimates show that it's even more, almost 15%. And there are a couple of reasons to this need. It's aging population, more and more individualized treatment, as we heard, more and more detailed analysis are needed on tissue samples. Also companion diagnostics, where the whole workflow from diagnosis to drug treatment is put together. And as I'm sure Martin would confirm, limited pathology resources with just not enough pathologists being trained, and the ones working now approaching or even passing the retirement age. So our technology is based on upconversion nanoparticles, and we are aiming to improve the IHC diagnosis. And we believe that our solution can be applied both to clinical use, this is our main focus, but can also be used for research, of course. And we are developing a 2-product system with whole slide scanner and a reagent kit. And the reagent kit is based on upconversion nanoparticles. And just to very briefly mention what they are made of rare earth minerals, usually erbium or thulium are used. And what is so special about these particles is that they are characterized by anti-Stokes effect. So they can absorb 2 photons of lower energy, but then emit 1 photon of higher energy. And what this means in simple terms is that we excite these particles with near-infrared laser, around 980 nanometers wavelength, but read the signal in 550 nanometers, for example. So whatever autofluorescence the tissue or counterstaining has in near-infrared light, we completely eliminate it because we read the signal in for example, visible light. So it's just such a big shift in excitation versus emission that there is absolutely no autofluorescence coming from this sample. And these particles have a lot of beneficial properties that can be used in imaging and digital pathology. So one is the anti-Stokes effect. They can be combined with H&E counterstaining because the particles are not visible in bright field, while hematoxin eosin would not give any signal in near-infrared light. So you can have both in one slide and then just decide, as I will show you in a moment, to combine them or view them separately. There's great potential for multiplexing because the particles have very narrow emission spectra. So there's no need for spectral mixing as you would normally have in standard fluorescent staining. Quantification is possible with these particles. You can read signal from 1 particle or 100 of them. We are not there yet, but we have data suggesting we can do it. They are very stable. The particles are nanocrystals, so you can image them for a minute or 10 minutes. You can have them in daylight. They are not affected by this. So they are very stable and can be stored for a very long time. The samples labeled with them can be stored in room temperature in daylight. And the workflow is the same as for any IHC labeling. So autostainer can be used. And they have very low signal to noise ratio, as you'll see also in some images, because we only see the signal from the particles. We don't see any background from the tissue. So to show you some examples, first is just a cell line, BT474, expressing HER2, breast cancer marker. And you can see this just hematoxin counterstaining in this slide, and then signal from our particles. And then this is how we can combine the signal in one slide. And the advantage is that it is the same section. So you can have 1 or 2 markers labeled in one slide and have the counterstaining as well. And then just choose to show one or both, or just toggle the opacity between them. Another example for our kind of proof of concept experiments, we focus on breast cancer. So again, you can see here just hematoxylin, the HER2 labeling, and combination that you can toggle on and off. When we compare this technology with standard DAB labeling, what I would like you to focus in this image is that with our technology, we only have the signal from the marker of interest. You can see that the rest is just black. So we see a very big advantage for AI analysis, for example, because in this case, you have to define how brown it should be to be counted as positive. You have to teach the algorithm to exclude the nuclei, to exclude the surrounding tissue, while in our case, you only have the signal from the particles. So this analysis is much simpler. Here is just another example of you can prepare heat maps using this technology. It's just we are looking to many things to find the optimal way to present it. So to summarize it, combination of labeling or staining with hematoxin and eosin and labeling with our particles is possible. We think it's a very big advantage where you can combine both in one slide. You don't have to view two separate sections side by side, but you can just toggle it on and off. And UCNP signal does not obscure cell or tissue morphology. So in case of DAB, you just have the brown labeling and you cannot remove it. You need a separate section with H&E. But with our technology, you can choose to have both on or just one of them. Our technology uses all the advantages of UCNPs, the anti-Stokes effect, very low signal to noise ratio. And the scanner we are developing supports bright field imaging and UCNP imaging, of course. And our solution is fully compatible with digital pathology. This is the only way forward that we see with this technology that is used for digital pathology. Thank you, and I'll be happy if you have any questions after the presentation.
Thank you very much, Christoph. Interesting, and especially this with the possibility to multiplex, which is becoming more and more important. The next presentation will be given by Jan Stenvang, who is the Chief Technology Officer at Scandion Oncology. Scandion Oncology is a company that focuses on developing drugs that target chemotherapy resistance mechanisms. And Jan will give a presentation called Clinical Phase 2 Biotechnology Company Targeting Cancer Drug Resistance. Please, Jan.
Yeah, I hope you can see my screen.
Perfect.
Okay, great. Thank you very much for the kind introduction, and I'm very honored and happy to be here to present Scandion Oncology. As mentioned in the beginning, I'm the CTO of Scandion Oncology, and I co-founded Scandion Oncology together with Niels Brunner in 2017 as a spin out of Copenhagen, University of Copenhagen and Saniona. And in 2018, we got listed at the Swedish Stock Exchange market, so we have a Danish-Swedish connection here. Our focus in Scandion Oncology is actually the big problem that globally 18 million people get cancer diagnosed every year. And the sad news is that 9.5 million of these are dying from cancer every year. And I tried to calculate that in Swedish numbers just to make it more clear to you how bad this is. In Sweden, every day 65 cancer patients die from cancer. That translates to around almost 3 cancer patients dying every 20 minutes. So it's very serious business we're talking about here. And this is our company disclaimer. And so all these people that die of cancer, it's estimated that 90% of cancer deaths are related to cancer drug resistance. So this is why we chose to focus on exactly this problem. Since no major company so far has focused exactly on that, we thought there was an open window here where we could really make a difference. So what are we doing? We're trying to develop first-in-class add-on drugs targeting drug cancer resistance. So what we like to think is that our drug, SCO-101, which is our lead candidate, can be combined with anti-cancer therapies, various kinds. And by combining these two, we will then restore the sensitivity of the resistant cancer cells. We have our pipeline currently targets around 60% of the used chemotherapies worldwide. And initially, we are focusing on colorectal cancer, pancreatic cancer, and breast cancer. I will come back to that in more details. We also think that SCO1 has the potential to quickly reach peak sales, since this is will be subscribed together with commonly used anti-cancer therapies. And we think it has the potential to also be used as a first-line anti-cancer therapy in combination with existing standard treatments. The mechanism of actions are enabling our company to target a wide variety of cancer indications, and potentially we can increasing the addressable market. So here's a cartoon showing how we envision and picture SCO-101 to work. Over here you see that you have the early stages of cancer, and they will be treated with chemotherapy or other treatments, but now we're focusing on medical treatment here, which is primarily chemotherapy in our case. We can see that in some cases, unfortunately, few resistant cells will remain, and they will unfortunately grow up and cause metastatic drug-resistant cancer cells. And at that point of time, you have very poor chances of being treated for any good. So how do we see it over here? If we take the lower one first, this corresponds to this one, where we have the metastatic drug-resistant cancer cells developed. Or we could have up here where the first line treatment, where we have a few resistant cell lines that, as we saw over here, can grow up. Then we can add SCO-101, and hopefully we can target all the cancer cells, all the resistant ones. So we will hopefully prevent cancer drug resistance from occurring. This is our vision. What about chemotherapy? Is that a big market? Is that a growing market? You can say yes to both. It's estimated that in 2024, the chemotherapy market will be $56 billion. That corresponds to almost 500 billion Swedish crowns. And as you can see, it's growing 12% each year. So it's a huge market that we come into. And you can also see over here where we have the estimated incident rates for US, that breast, colorectal cancer, and pancreatic cancer are among some of the bigger ones. So we think we are addressing huge unmet medical need into a very large market of chemotherapy, into some severe indications that have many patients and have a strong need to be helped. So our pipeline is shown here, and as I said before, colorectal cancer, pancreatic cancer, and breast cancer is our initial focus points. And as you can see up here, SCO-101 is already in phase 2 clinical testing. I will show you the details in a minute. And in October this year, we also initiated a new clinical study in pancreatic cancer. I will also tell you about that. And we have plans to look more into a breast cancer study here that we will decide the exact design later. However, we also have solid tumors of other kinds with another drug in our pipeline called the SCO-201 that is different from 101, obviously. We have 301 that is targeting other cancers again, and then we have a huge library of analogs. We have 700-800 analogs, and these we are using for preclinical screening. So let me tell you just very briefly in details the two studies that we are doing. Our colorectal cancer studies is comprised of two parts. You have part 1 over here. This is with 12 to 18 patients. This is a part of the study that is focusing on safety, toxicity, and also, of course, the effect of the treatment. This is meant to find out how much SCO-101 can the colorectal cancer patient tolerate in combination with FOLFIRI, which is the chemotherapy we combine with. Then we will select the dose, and then we will increase the number of patients over here in part 2. And then we will have a proof of concept study where the primary endpoint is the efficacy of the treatment. You have the timelines down here, and we can go through them in details if you wish. This is our pancreatic cancer clinical study that we have just initiated. And this is also based in 2 parts, part 1 over here and part 1. However, you should know that this is fundamental different from the colorectal cancer that I just showed you. It will also have two parts. Yes, that's true. But this will be combined as a first-line treatment in pancreatic metastatic pancreatic cancer patients with nab-paclitaxel and gemcitabine. So the colorectal cancer was last-line treatment. This is first-line metastatic treatment, and we will have select the dose again. And then in part 2, the study will also be different from the colorectal cancer study, because here in the pancreatic cancer study, we will have a randomized trial with the patients receiving SCO plus nab-paclitaxel and gemcitabine, and the other arm will be the standard treatment of nab-paclitaxel and gemcitabine. These designs allow us more to look at time measurements like progression-free survival, overall survival, and so forth. So colorectal cancer, last-line treatment; pancreatic cancer, first-line metastatic treatment. So how do we go around finding new drugs in Scandion Oncology? Yeah, we have a drug screening platform that we have developed, and it consists of drug-sensitive and drug-resistant cell lines. So we can compare these two, and we can screen for new drugs that is able to target the drug-resistant cell lines. Then we will put these drugs into our pipeline. We will, of course, thoroughly do preclinical studies and validate the efficacy in preclinical models and the mechanism of action. And then, if that comes out successful, we will move on to the clinical studies. And eventually, when we have proof of concept, we will look more into partnerships and licensing. This is just to give you an example of the field and the deals that have been selected for this presentation, but I think I would like to draw your attention only to the lower one. This is an American company called ORIC, which also has a focus of drug-resistant cancer. They have different molecules, different indications, and different mechanisms of action. But what we think is interesting is that their market cap is more than $1 billion US. So this is telling us something about the interest and the size of the market. So just for the investors, we think that we have 3 highlights for the investors. So we target drug resistance. We can reach a rapid peak sales by combination therapy with SCO-101. The market we're targeting is huge, US $37 billion. We have a well-documented mechanism of action, and our lead candidate is currently in clinical phase 2 trials, and we have started the metastatic pancreatic cancer clinical trials in October. Our management and board of directors are, I would say, extremely experienced and capable in the field of biotech and merger and acquisition. And I'd like to show you just them as the last slide. Some of them you may know. Our CEO, Bo Ode Hansen, was attracted to our company starting as a CSO of 1st of October from Geneva, US. Niels Brunner has been the former CEO, is now the CSO, also co-founder, and has also been establishing the Oncology Network here at MVA. And then we have CFO, CRO, CMO, and CCO. In the board, I think you should pay attention to Peter Høngaard, which is called Mr. Biotech Denmark, has a proven track record, as you can see here. Carl Borbæk, most of you probably know in Sweden, and he's also one of the founders of the Oncology Network, together with Niels. And then we have Thomas Feldhus here, who has been the former CFO of Saniona, who's also listed in Sweden. So I think we have a very capable management and board of directors. So with these words, I would like to thank you all for your attention and looking forward to your questions. Thank you very much.
Thank you very much, Jan. It's impressive to see what you and your company have achieved in a rather short period of time. And now for the final presentation, which will be given by Erik Steinfelder, who is Biobanking Market Development Director at Thermo Fisher Scientific. And Erik will give a presentation called Public-Private Partnerships, a strong way forward to maximize impact. Please, Erik.
Thank you very much, Johan, and welcome everybody to the last talk that I'm willing to give. I always need to know which slide you are seeing. Are you seeing the right one, or do you see presentation mode?
Presentation mode is fine.
Yeah, it's fine. Okay, thanks very much. Yeah, I would like to use the last 10 minutes of the presentations to discuss and share a little bit more about public-private partnerships. As said, Eric Steinfelder, I'm the Biobank Market Development Director within Thermo Fisher Scientific. And I would actually like to use biobanking as an example how public and private could work together, which obstacles we had in the years past, but what kind of nice opportunities we have ahead of us, and how we could work together in a more efficient way. So like I said, I would like to use it a little bit and build it a little bit around biobanking. Active since 15, 20 years. We learned a lot in the last decade, and especially on how to build, how to use things, how to be, basically shift from a sample storage facility to more a service provider, because that's at the end what you would like to do. You have those collections, you have really high valuable data, but how can somebody actually make use of it? And I think around 15 years ago, UK Biobank was starting up, and maybe some of you know it, Biobank Graz. In China, we had the Kadoorie Biobank, large population-based biobanks that still are being used today, build their collections, but also help industry, help other researchers to actually move their scientific research forward. But in the past, we had lots of discussions within the biobank community before we were ready to actually look at industry to partner or to look at other academic centers around the world to start talking about collaboration. And one of the biggest issues was not only the discussion, is it a biobank or a biorepository, but it was really around quality. Do we actually know what we store? Do we have data to back up that what we've stored in the last decade is stored under the right conditions, always had the right temperature, wasn't handled in a bad way? You need to have the data. But then always the question was, yes, but what do you mean with quality? It can be fit for purpose. I think, and I will discuss a little bit later on, we've overcome those discussions. And I think we've passed the time that people only think that a biobank is just a selection of freezers with some samples in it. I think biobank community reached out to other organizations, other bodies, and connected nationally. I know there is a strong national Danish biobank network. There is one in Sweden, there is one in Finland, but also on a European level. There has been efforts to connect and to see what can we do amongst each other to make sure that we do the right thing, that we learn from each other's best practices, and that we guarantee that we actually have a process towards standardization, which in the beginning was quite a challenge. Last but not least, we started working closer and closer with patient organizations. What do they need? Because they are providing their samples. They are giving consent to actually use the data. But do we use it in a proper way? Does it help them? We learned a lot, but we got there. I think it's good to see the improvement that we made. But just going back to quality, I think that was a big issue. And I've spoken to many researchers that said, Eric, I know you're a fond of biobanks, but if we don't know what they have stored, how they have stored it, and if there is no data to back it up, I'm not sure if we can use it. And to back that up, actually, a German group of researchers did a survey in December 2019, where they asked hundreds of researchers, if you had to work with a biobank, what would be your main criteria? And again, there it was high quality, reliable storage conditions. So that's really, really mandatory. So it's helpful that there is now an ISO standard. Standard is there since October 2018, and it helps biobanks not to reinvent the wheel again, but really look at processes that we can standardize all around the world and say, hey, this is the way we did it, and that actually makes the work that we're doing comparable. And not only on the sample side, but also on the data side. There are now initiatives around data provenance to see what is actually the source. Is this the information that you need? Can you guarantee that also the quality of data is good? One of the few initiatives that really helped was the European Research Infrastructure, BBMRI, ERIC. They did a colorectal cancer cohort where they said, let's go to 25 biobanks in 12 different European countries. Can we bring them all together into one big database What kind of ethical and legal issues will there be? Will there be quality issues, etc.? It took 2.5 years to finalize that, but there is a blueprint available. With that blueprint, you can go external and say, hey, we got a great set of data. If you're in oncology, if you are in research, we might be able to help you. So you would say they are sitting on gold and they have a very high valuable collection. But then we still see, if you talk about partnerships between, for instance, biobanks and other academic institutions or industry, there is still a gap in understanding, in communication, and misunderstanding on both sides. We see a lot of biotech companies looking for samples, really looking desperately for samples that biobanks have, but sometimes simply a specific type of data set misses. The quality cannot be guaranteed. There is no history on the storage conditions. But also, it's not always a research question according to the Biobank Ethics Committee. If you are in industry and you are working in medical devices, if you want to launch a new product or launch a new assay, you need to prove that it's working. You need to prove, of course, that it's working and confirming what you are trying to claim. But therefore, you need samples. You need samples to prove that. But for a biobank, they say, well, that's not a research question. So if you don't have a research question, it is not going to result in a paper. I'm not going to share the material. So there is a high demand on one side. There is a high availability on the other side. The parties don't get together closer. And that's where I think we should be able to do a little bit more and see how can, from a biobanking perspective, academic, private and public sector get closer to each other. There are legal barriers, there are ethical questions, but they should be overcome. It's not undoable, but it just takes time. And that's another challenge. We see in industry that when people ask things, 2-3 weeks is maximum time they were willing to wait. We see that a biobank thinks that they are very fast if they answer within 3 months. So these simple things, if we can get that out of the way, it might be easier to get closer together and to work more together, because there are so many opportunities ahead. We do see a good future for public-private partnerships in that area, and especially when you look at oncology research and what the funding is available in the months ahead and in the years ahead. And one of the things that really helped actually was the search for positive samples around COVID. When the pandemic started, nobody really knew what was going on. Everybody was looking to understand what is the disease, what is going on, what is the effect, how does it work from an infectious perspective. Everybody was looking for samples. Everybody was trying to connect with biobanks. And they were very fast in making sure that they had the samples, but also had the right data. So they could share treatment protocols. They did have information around clinical symptoms. They could say, hey, this was the disease severity, the duration, and the outcome. That became very powerful. The other hand, which made biobanks really valuable for pharma and research side, is that sometimes they could go back to the population that was positive, but they had samples from a year earlier, where these people were still healthy. And then you can make comparisons. And then you have a really, really rich set of data. And one of the options that they made now is that there is a catalog available where BBMRI, Eric, and ISBER, two biobank organizations, made a catalog where you can look online, search for biobanks that have positive COVID examples as samples. So there you see it's a first step to open up more and also have the data to back up. This is how we can help you, and this is how we can drive your research. And it certainly helped us as a company, because we wanted to launch a new allergy test around COVID, a fast test based on the platform that we already had. And also there, if you want to make sure that this test is valid, it's working, you need to come up with proof. So there we had the support of several biobanks in Europe that could help us out. And it was really, well, without them, we couldn't have done it. So good for them. Other partnership that I wanted to share, looking at the time, was Conception. That's where a pharmaceutical industry said, hey, we would like to know more about certain drugs and the effect on pregnant women. Project Conception is paid and driven by a consortium of academia and pharmaceutical industry, where they look into the details of it. Because this very strong private and public network was there, they said, well, if you're doing this anyway, can you also look at the impact of COVID to pregnant women? So they used the consortia already there, and they used the strong connections that academia and the pharmaceutical industry already had. And I think that's powerful, but it's also the way forward to actually make use of high valuable data collections and samples that are out there. Just to give you an example, we have around 100 million samples stored in Europe, and we use less than 5%. So we could do a little bit more to drive the research. FinGen, other projects. Maybe I could touch this one as a last slide. The EU mission on cancer, you might have heard it. There's going to be a lot of initiative and a lot of funding available to meet the target in 2030, where the Commission wants to save more than 3 million lives for people that are diagnosed with cancer today. That requires a lot of research, that requires a lot of scientific expertise. This can only be done by good work together from policymakers, academia, industry, regulatory bodies. So I think that's where we as a company can play a part, but also with other stakeholders, we could have a strong case. It's good that the mission board has a wide set of stakeholders involved, including the biobank community. Yeah, and I really look forward to do a little bit more in that. Last but not least, then we also have to look at the position of the different parties involved. Sometimes it's not always clear what they can bring to the table, what is qualifying them to be a partner, and how can they be a successful partner. I'm running out of time, but there is a link in this presentation to a poster on this topic. But in summary, we cannot do it alone. We have to work together. And the more we can facilitate public-private partnerships, I think the better for the research and ultimately for the patient. Thank you.
Thank you very much, Eric. Interesting. And with this presentation, we have time for a short panel discussion. We have received a couple of questions, and I would like to start by asking Eva. There are thousands of articles written about cancer and genetics, but all this information, it's a very small fraction of it that's used in routine healthcare. What needs to be done to integrate this information so it benefits the patients?
Thank you for the question, Johan. I think that one of the things that obviously needs to happen is that it needs to be commercialized, and also it needs to be commercialized, and also better algorithms for actually calling the mutational signatures that can be used. This requires a lot of clinical validation, obviously, as well. So I think there's still quite a way to go, but I think one of the ones to start with is actually microsatellite instability. I think that's a very good mutational signature. I think it's also very valuable. We know a lot about it clinically, diagnostically as well. So I think starting there and then building up from there. The BRCA1, BRCA2 signatures is another one where we also know quite a bit, and that it's very important for, say, treatment with PARP inhibitors. So I think we have places to start where we can start integrating and then exploring either into new cancers with similar signatures, and or of course also discovering new signatures in cancers we know quite well.
Thank you very much. And then we have a question for Jan. You represent a company that is, it's a very young company. What have been the major obstacles for you when you have built this company?
How long time do I have to answer that? No, obviously there has been many obstacles. I think the traditional ones are, of course, raising capital. That was the first one. Second one is to attract the right people with the right knowledge, which was, I guess, the second one. But basically, after attracting or completing these two tasks, then I think it has actually been relatively straightforward. That's maybe a little bit blunt to put it like that. There's been many obstacles still, but I think once you have the money and you have attracted initially the best people with the right competences, and then I think actually you're in a good position. But obviously, you know, IPR is an issue. Conducting clinical trials is a major issue. And you know, keeping track of an organization that is growing is a constant challenge that we constantly are improving at Scandion Oncology and constantly building on that. So I guess we have been through many of the traditional challenges, but I think we have managed to actually overcome them in a reasonable way and in a quite fast way also, I would say.
Where do you find the right competence? How do you recruit scientists?
Yeah, that has initially it has been through, you know, the network. Who do you know, and who do you know that knows good people? That was our original approach. But as I also mentioned here in the beginning in the presentation, that we attracted a very competent new CEO October 1st, and that was, you know, a professional approach, screening for competent candidates and having interviews with these. So that was a very professional approach to actually attract the right CEO that will take us, you know, through the next phase in the company life cycle.
And then I have one question here for Eric. In Europe, I guess there are, I don't know, 500-600 different biobanks.
Yep.
Most of them are never used.
Yes.
What needs to be done to start using? I mean, there are lots of people who collect samples, but they never use them.
Yeah, it's a big issue, and I think that there are a few reasons behind it. One is simply that people are so fond of their collection that they basically call it their precious, and they don't want to share unless it's within their own little world or little center. The other thing is that there is a mismatch with what the researcher or industry really wants, and then they can deliver 85% or 90%, but it just doesn't fit. So then people move on and try to find somebody that has 100% fit. So we also try to educate the biobanks, really look at a broad spectrum and collect as much data as you can with what is allowed within the law, to make sure that you are attractive as possible for other people to use your material. Last but not least, I think biobanks are doing fantastic stuff, but they're really shy. And if nobody knows that you exist, you have to go out there and say, hey, this is what I have. I'm unique. I can help. I can drive your research. What can we do? But that's not in their nature. Yeah.
So, Christoph, where do you're working for a private company? Where do you find samples?
Well, it's a very difficult thing to find them, and we just have to buy them from different suppliers. This is not easy. The quality is often not very good. So we actually find it very difficult to find the right samples. The only way being just finding the best possible supplier and buying them.
And finally, one more question there for Yuri. I mean, blood circulates around all the organs, so it would be nice if you could measure everything in blood instead of taking biopsies and looking and using microscopes. What needs to be done to diagnose and screen for different diseases using blood instead of the actual tissue?
Okay, so I think what you need to do is to address some that Eric brought up, actually to connect interaction, and I would say both on private industry, pharma, and also academia and healthcare. The other aspect, I think, I mean, right now you have central laboratories in hospitals, right? I mean, you do have a basic technology platform that you are doing. And I mean, if you take southern Sweden, you do like 10-15 million analyses a year in all disease areas. Now that's straightforward, very often antibody-based assays. I think what you also have is, if you look to probably mostly in North America, US, you have a lot of new platforms and technologies that are taking off. Startup companies, you can actually take a small dot, and then you can ship it off and get analysis of different proteins as such. I think that also new platforms, like if you look to Cedars-Sinai, Los Angeles, or in other big centralized hospital in the US, insurance companies, clinical companies, there are now being used in order to screen also for patients in different type of diseases. So I think technology will drive it together with the medical side, but I also think that there are available right now platforms within central units in healthcare, probably multiplexing to see more than one thing in one go. And you only need a minute amount of blood, actually. A drop is enough.
Okay. One final question for Jens Victor. Short answer, please. What is the biggest mistake that your clients do when they come to you?
Well, thank you very much. It actually brings me back to the first question you asked, Eva, about why do all the published findings not reach the clinic? And that's because they are published before they are patented. So actually, publishing findings can prevent that they are used in practice, because you will not get investors on board unless you have filed a patent application first. You will not get pharma to invest in your research if you have published it and shared it with everybody. So sharing research with everybody can actually prevent its implementation in the clinic. It seems counterintuitive, but it's actually sometimes so.
Finally, I would like to thank all the speakers for excellent presentations. I hope that this has paved the way for new collaboration and provoked some ideas. And thank you, all of you in the audience. And I would like to give the word to Petter for a few final remarks. Please, Petter.
Thank you so much, Johan. I would just say, just as you said, Johan, a big thank you to all of you who followed us this afternoon. I hope you have enjoyed. And also from our side at Medicon Valley Alliance, a big thank you to the speakers, and especially to you, Johan, who has guided us through this event. It has been a true pleasure, and I hope to see all of you soon again. And we will, of course, continue the discussions on how we can increase the collaboration across the Sound and build on the private-public collaboration tradition that we have in this area, not least. So thank you once again. We will send out, as I said in the beginning, a follow-up email tomorrow. Please answer the short questions and survey that will be included. That will help us a lot. Thank you so much, and take care, everyone, and see you soon again.